Hello everyone and welcome to our MycoCosm case study workshop. My name is Arthi Inagandla and I'll be walking you through our case study today. So first, a little bit about me. I'm an incoming senior at UC Berkeley, where I'm doing a major in molecular and cell biology and minoring in CalTeach or science and math education. I'm currently doing an internship through CalTeach and the Joint Genome Institute at Lawrence Berkeley National Lab, where I'm doing research using the fungal group's data portal, MycoCosm. What is the Joint Genome Institute? The Joint Genome Institute operates under the Department of Energy, and its goal is to advance genomics research related to environmental characterization and cleanup, and provides genomic analysis data to the public. The way it does this is through its multiple different data portals, including a fungal portal, an algal portal, and a microbial portal. The one that we'll be focusing on today is the fungal portal, called MycoCosm. So the DOE focuses a lot on bioenergy, but what is bioenergy? Bioenergy is a way to use biofuels from sustainable sources of biomass to alleviate our dependence on fossil fuels. Today, we'll be exploring one way fungi can help us access plant biomass to eventually use for bioenergy. So what does MycoCosm look like? We're going to go to the data portal and interact with it right now. This is what the MycoCosm homepage looks like, and it shows an interactive fungal tree of life. So if I click here, I can see that we house over 2,000 fungal genomes and counting. And so now I'm going to be showing you how you can interact with the fungal tree of life and, explore any different fungi that you'd like to examine today. So we're going to be focusing on specifically Mycotina. So Mycotina houses two types of fungi that we're particularly interested in, white and brown rot fungi. So before we do that, and before we learn more about white rot and brown rot fungi, we're going to be looking at something a little bit more recognizable. So if I click on Mycotina, it shows me this drop-down menu of many different things, which we'll be exploring later, but we want to look at all of these fungi that are available to us. So this shows us all the fungi that are specifically in Mycotina. So if I don't know a fungus that I want to look at, I can simply just scroll through this drop-down menu and look for one that I want to look at. So we're going to look at Amanita muscaria. And I want to know if you guys can guess which fungus or which one this is based off of in popular culture. So you might have guessed it, you might have not guessed it. Amanita muscaria is the most widely recognizable fungus in the world, and it's featured in Super Mario Bros. As Toad. So the great thing about the MycoCosm data portal is it doesn't just show you important scientific and biological information about this fungus, but also shows you important and fun tidbits about the mushroom itself. So now we're going to go and interact with the fungal portal a little bit more and explore what white rot and brown rot fungi are. So we're going to again go through this specific family here, Mycotina, specifically Agricomycetes. So again, if I don't know the name of the fungus that I want to look at, I can simply just scroll through this drop-down menu and look for the fungus that I want to look at. This one that I want to look at is Phanerochaete cryosporium, and since all of this is in alphabetical order, I can simply just scroll down to P and then find the one that I want to look at. So this one is right here. I'm just going to click on it. And again, this is going to take us to the info page of this specific fungus. So now this is telling us that Phanerochaete cryosporium is part of a group called white rot fungi, and this produces oxidative enzymes that degrade lignin. So what is white rot fungi? What is lignin? These are all questions that we're going to be exploring in a little bit. So just again, to clarify, there are scientific papers that you can look at over here. There's more information that you might want to learn about this specific fungus. So now what if I knew the name of a fungus that I wanted to look at and I didn't want to look through all of this, tree because I didn't know where it was located? I can simply just search it in the search bar. So if I know that I want to look at cornea, I can just enter that in there, search in MycoCosm, and it'll pop up over here and I can just click it similarly how I did earlier. And this will take me to the info page of this one. So now we can see here that cornea produces a brown rot in which lignin is not degraded. So again, what is lignin? What is brown rot? What is white rot? These are all terms that we're going to be defining very, very soon. So just to conclude on this MycoCosm info page and homepage tutorial. There are so many different genomes that you can look at, so many different fungi. All you have to do is just click around and read and learn more. You can read the research papers if you'd like to or not. It's really up to you, and there's so much information that you can look through. So now we're going to go back and look at exactly what white rot and brown rot fungi are. So thank you for joining me on this exploration. What is the focus of our case study? Today, we'll be exploring white rot and brown rot fungi. And before we answer our question, we need to understand what brown rot and white rot fungi are and how we can distinguish them. So this is what brown rot and white rot fungi look like on decaying wood, which is exactly what they do. And you can see that it was named specifically after what it looks like. So this is brown rot clearly, and this is white rot. But that's not enough. We need to know exactly what the specific differences are that make them what they are. So just to give you some background, what is this case study based on? This case study that we'll work through today is based off of a research paper called Extensive Sampling of Basidiomycete Genomes. And this was written by Robert Riley and published in 2014 in PNAS, a scientific journal. So many of the questions that we will be asking and then answering today are really based off of the fundamental questions that were asked in this paper. So we definitely have these authors to thank for giving us the basis of our case study walkthrough today. So now let's examine the differences between them. This picture on the right shows 3 cell wall components of a plant cell. If you remember, the plant cell wall has many different functions, one of which is to keep the plant upright and act as the skeleton. This is what lignin, hemicellulose, and cellulose do, among other things. They provide structure for the plant. So white rot and brown rot fungi each have different enzymes that target different plant cell wall components. White rot completely degrades lignin, and brown rot is unable to degrade lignin. Brown rot, however, completely degrades hemicellulose and cellulose very rapidly. White rot has the ability to degrade some hemicellulose and cellulose, but does so very slowly. So if you remember back to our picture over here, you can see here why each of them are named brown rot and white rot. So if you remember what I had said just now, brown rot is unable to degrade lignin. So this is what you're seeing here. It's the lignin because it has degraded all the hemicellulose and cellulose. So white rot is able to degrade all 3 components. It degrades lignin very quickly, but it does degrade hemicellulose and cellulose very slowly. So what you're seeing here in white is really the hemicellulose and the cellulose, or the sugar. So that's an important distinction to make, why these look the way they look. So brown rot, like we said, does not have the ability to degrade lignin, and white rot does have the ability. So now we have to ask this question: is this a strict dichotomy? Meaning, are white rot and brown rot completely separated? Are the enzymes that act on each, relegated to only each one, and not, is there any overlap? And the answer is no. The research paper by Robert Riley examines how certain white rot enzymes that were previously thought to be specific to only white rot were also present in brown rot fungi. So this is what we'll be exploring today, is that the fact that what enzymes are specific to each one, and also how this might not be a dichotomous fact. So now our case study question: how can we use MycoCosm to explore enzyme and protein differences between white and brown rot fungi? So here we have the 4 fungi that we'll be looking at. First one is Auricularia subglabra, fungus A for ease. This is a white rot fungus. Second, Calycera cornea. We had explored this in the MycoCosm homepage. This is fungus C, or a brown rot fungus. Next, Phanerochaete cryosporium, fungus P, white rot over here. And then lastly, Gliophilium trabeum, fungus G, a brown rot fungus. So we're going to be looking at these 4 fungi, 2 white rot, 2 brown rot, and see what are the protein and enzyme differences between them. So we're going to simply click on this link. You can enter it into your browser page and explore it for yourself after we are done. So now this takes us to the home page of this comparative approach, where we're looking at the differences across these 4 ones, and it has been created specifically for this case study. So when we look at the enzyme families and protein domains, we will only see comparisons across these 4 fungi. So now I'm just going to briefly explain what this menu bar up here says. So today we're going to be using two of the things on the search bar, the annotations right here and the MCL clusters. First, we're going to talk a little bit about the annotations. Genome annotation is a process of annotating the genome, or looking very closely at the nucleotide sequence to find proteins, coding sequences, enzymes, transcription factors, etc. So when these are found during genome annotation, they are put into MycoCosm for easy access for users. So we can look at the annotations and explore what they mean, especially across different enzymes, different organisms as well, for a comparative approach. One of the drop downs for the annotations that we'll be using are the Kzymes page. So just to point out, there's a help button here that is present at every single menu bar, and it can just show you how to explore the data portal, and it'll help a lot if you're confused on how to navigate. So now let's go to Kzymes and see what's going on. So what are Kzymes? Kzymes are carbohydrate active enzymes. These enzymes might synthesize or break down carbohydrates. So this is of interest to us because we know that we are looking at carbohydrate molecules today from the plant cell wall, because we know that white rot and brown rot fungi act on different portions of the cell wall. So the first family that we'll look at are auxiliary activities over here. So auxiliary activities family contains peroxidases that mainly act on lignin. Peroxidases are enzymes that catalyze oxidation reduction reactions. So when we click this right here, we're going to get a drop down of different subfamilies. So the first subfamily that we're going to be looking at is AA2. Based on previous research, we know that AA2 is a peroxidase that breaks down lignin. Since we know that white rot breaks down lignin, and there are 19 and 16 copies in fungus A and P, both white rot fungi, we can confirm the fact that fungus A and fungus P are white rot, while fungus C and fungus G are brown rot, since they don't possess any copies of the enzyme subfamily that breaks down lignin. The next subfamily that we're going to be looking at is AA9 right here. AA9 has a different type of peroxidase that may oxidatively act on lignin, but here we see something a little different. We see similar numbers, 19 and 16, in both fungus A and P, but we see 4 copies in fungus G, which is a brown rot fungus. So this is obviously a little weird. However, as we discussed before, white rot and brown rot fungi don't exist in a dichotomy. And there are instances in which brown rot fungi might possess enzymes that we previously thought were typical of white rot fungi. So this enzyme family also has a role to play in brown rot fungi, and this is the perfect example of that. So we're just going to collapse this and look at our next Kzyme, which is the CBM, carbohydrate binding module family. So we're going to be specifically looking at CBM1 right over here. So CBM1 domains in white rot specifically perform targeted binding to cellulose to increase efficiency of degradation by enzymes, by a group of enzymes specifically called glycoside hydrolases over here in the GH group, specifically GH6 and GH7, which you can explore more on your own later. But over here in the CBM1, we see that white rot fungi, fungus A and fungus P, have greatly increased domains or copies of this specific CBM1, greatly elevated. But we can see over here in fungus C and fungus G that there are only 1 copy each. So this gives us even more confirmation that the known function of the enzyme families that we are looking at match up with the number of copies for white rot and brown rot fungi. So now that we have confirmed which ones are white rot and brown rot fungi, A and P are white rot, C and G are brown rot, specifically through enzyme family exploration, we can now back up and ask a reverse question. Which genes are present in either group, white rot and brown rot? Or which genes make either one unique? So that takes us to the MCL clusters page, right over here. So the MCL's cluster page shows us the result of protein clustering that is depicted right over here. We can basically compare protein sequences with each other and group them into bins of similar sequences called clusters or protein families. The colors show proportion of domains found in that cluster, so blue, and also the thickness of the donut shows abundance of that domain relative to the largest full circle, for example, right over here. Columns indicate organisms, and rows show protein clusters. With the number of proteins each organism contributes to a cluster. So over here, you can see that there are 177 of this specific one distributed across these four, compared to 115 of the purple one. You can see that it is only present in this and this. So this is a really great way to see what domains are present in the organisms or fungi that you're looking at specifically. So right now this page is showing us clusters present in any of these fungi, but there is a way for us to look at clusters only present in the white rot fungi, for example. So we want to know what's unique to the white rot fungi. And the way that we're going to do that is we're going to change up the settings over here. So right now they're set to any, but we want to know what is unique to A and P. So the way we do that is we change A and P to +1, which I'm doing right now. And then we change the C and G to 0 because we don't want to see what is in the brown rot. We want to see what is in only the white rot fungi. So this shows us everything that's unique to A and P. And the first thing that pops up is this peroxidase family. So what does that mean? So if you remember, we are seeing very similar numbers to what we saw in the AA2 enzymes for the Kzyme page. This is because this family of peroxidases include the AA2 enzymes, but it's important to note that not all members of this peroxidase family degrade lignin and may have other functions. The MCL clusters page has so much information to show us. We can also look at these domains specifically that show us glycosyl hydrolase family 7, as well as the fungal cellulose binding domain, which correspond to the glycoside hydrolase family that we said degraded cellulose in white rot, as well as CBM1, the binding domain for cellulose in white rot. So if we explore more here, we can see many different things, but what we want to know are the domains, so where they are located. So as you can see here, all of these contain the glycosyl hydrolase family 7, and most, if not all, contain the fungal cellulose binding domain. So these domains are color-coded based on the protein shown. So as you can see here, this corresponds to the glycosyl hydrolase, and this corresponds to the fungal cellulose binding domain. So as we can expect from their function, we can see by this model they occur together for the most part. And also we know that these are specific to the white rot fungi. So it's really a really valuable tool to see, what we might want to explore of particular protein domains and really what sets apart white rot and brown rot fungi. So now this is the end of our MycoCosm exploration of the differences of white rot and brown rot fungi. Now let's wrap up. So conclusions. What did we accomplish and why is this important? So first, we used the Kzyme browser to understand how we can take known enzyme families that act on different parts of the plant skeleton and investigate the differences in number between white and brown rot. So we saw this in the AA2 subfamily of the Kzymes. However, some brown rot might actually have enzymes that white rot typically have. We saw this in the AA9 subfamily. We can also use the MCL clusters page to work in a different direction and find which domains are present in the white rot specifically that the brown rot don't have. And next, I wanted to point out, sometimes concepts in science don't always fit neatly into two boxes. Oftentimes we'd like them to, but the reality is that more often than not, they don't. With more and more research and exploration, things are more likely to be a continuum rather than two separate things. And lastly, what does this mean for bioenergy? If you remember from earlier, hemicellulose and cellulose are sugars, and although white rot is able to degrade hemicellulose and cellulose, it does so very slowly, essentially fully degrading the lignin and leaving behind the sugars. With the sugars of the plant left behind, this can be used for bioenergy. Bioenergy, just to remind you, is a way to use biofuels from sustainable sources of biomass, in this case, hemicellulose and cellulose, to alleviate our dependence on fossil fuels. So understanding what enzymes white rot fungi use to degrade lignin is really important to eventually using cellulose as bioenergy. So this is why exploring these differences are so important to us. Now, next steps. So what can you do with all this information that you've learned? So for the Kzymes browser page, you can repeat the same exploration with different enzyme families, such as GH6 and GH7, as well as explore the rest of the Kzymes shown. Some questions to think about: what do you see that is unique to the white rot and brown rot fungi that we identified, and what might be surprising about some of the numbers that you're seeing? Next, for the MCL clusters browser page, you can instead search for gene families absent in both white rot fungi, or find gene families absent in white rot fungi but present in brown rot fungi. So you would set the fungus A and P to 0 and set the brown rot fungi to +1 to see what's unique to the brown rot fungi. Then you can explore the models and domains associated with them. And lastly, go crazy. As I said before, MycoCosm is a free resource with so much data for you to explore and play around with. You can explore any different fungi or fungus that, you know, sparks your or piques your interest. Explore the MCL clusters page for that one. Play around with the Kzymes. You know, there's so much stuff for you to do, and I really, really encourage that you take advantage of this opportunity and, you know, really learn more about what it means to conduct research and explore scientific data. And thank you so much. I appreciate you listening to me talk about this case study and walk you through it. Hopefully you've gained a lot more information about how cool fungi are, and second, how applicable they are to real world use. So if you have any questions or comments, please contact me at my email here. And if you also want to contact any members of the fungal group, you can contact them here as well. So thank you so much. Have a great day.